Hey everyone, and welcome to your Fluid, Electrolyte, and Acid-Base Balance 1 lecture, also known as Fluid Balance 1 lecture. It's less of a mouthful. But this is going to be your lecture recording. I will break it up into 3 separate videos so that they're easier to digest. And one thing to remember is I am going to show you the case study for this lecture, but remember that you are only to submit a case study if I assign this lecture to the entire class online. If you're absent when we have an actual in-class lecture, please do not submit a case study because case studies are used for attendance. And so if you were not present for an in-class lecture and you're just watching this to catch up, do not submit a case study. So just wanted to say that, and let's go ahead and get started. Okay, I think you should be looking at the case study now. So this case study number 83, Austin, a 19-year-old male, was rushed to the ER due to seizures that led to a coma. The patient's friends reported that the seizures began shortly after he completed a dare to ingest over a quart of teriyaki sauce. The patient did not respond to anti-seizure medication, but began slowly improving after being administered a water/dextrose, which is a sugar solution, via nasal tube. And so what is your diagnosis? All right, so fluid balance 1. We have lots of things to go over today. We're talking about the 3 fluid compartments of the body, which should be a review. We'll talk about the solute concentration and osmolality of each compartment, which we've started. We're going to add some things onto that today. We're going to contrast the overall osmotic effects of electrolytes and non-electrolytes. We'll describe factors that determine fluid shifts in the body. We'll list the routes by which water enters and leaves the body. We'll talk about feedback mechanisms that regulate water intake and hormonal controls of water output in the urine. We'll talk about what are called obligatory water losses. We'll talk about causes and consequences of dehydration and hypotonic hydration, which is over intake of water. Then we'll indicate routes of electrolyte entry and loss from the body. We'll describe the importance of sodium in the body's fluid balance and electrolyte balance, and we'll describe the mechanism involved in regulating sodium balance, blood volume, and blood pressure, and explain how potassium, calcium, and anion balances in the plasma are regulated. Okay, so the body fluids consist of water and solutes in 3 main compartments. And so you should already be familiar with these compartments. So we have the intracellular fluid, or the ICF, which is the fluid inside of our cells. Now, the fluid inside all 3 of these compartments constitutes or accounts for 60% of our body weight is the fluid in these 3 compartments. And then of that 60%, 40% of that is from inside of our cells, because we have trillions of cells. And so, or I think maybe equal to a trillion-ish, but billions at least cells. And so 40% of that 60% is intracellular fluid. Another 20% is the extracellular fluid. Well, I lost my spot. The other 20% is extracellular fluid, which is subdivided into our interstitial fluid, which is the fluid in between our cells, and then our blood plasma. And so now when we say that total body water is about 40 liters and about 60% of our body weight, that is a metric given for an average healthy adult male. So these numbers are going to be a little bit different depending on your age, body mass, percent body fat, and your sex as well. So some examples, and I believe all these examples are in your lecture notes, but I'll go ahead and write a couple of them out. But again, this is just for a healthy average adult male. But if you're an infant and made up of a lot of cartilage and not so much bone, infants, and like I said, I think this is your lecture notes. Let me see. Yeah, so infants are about 73% water. I think your lecture notes round it to 75%, which is totally fine. Young men, as we see here, are about 60% water. And then young women are more like 50% water. And the reason why women have less water than males is because fat is not a very hydrated tissue. Fat is only about 20% water, while skeletal muscle is about 70% water. And so females in general have more fat and less skeletal muscle than the average male. And therefore, they're going to be made up of less water since fat is not really a hydrated tissue. Skeletal muscle is a very hydrated tissue. So if males have more skeletal muscle mass, they're also going to be carrying more water than the average female does. And so in general, the more muscle you have, the more water that you're carrying. And that can be balanced between those three fluid compartments. Okay. Something else to consider. My slides to keep going. Okay, here we go. Here are our three fluid compartments, and let me see if I can erase this stuff that was here. There we go. I haven't used Zoom recordings in a while, but here are three fluid compartments. You should already be familiar with this image here. We talked about the concentrations of sodium across these three fluid compartments, 12, and then the units for these are in milliequivalents per liter. We'll talk more about milliequivalents per liter and that measurement a little bit later. So I'm not going to write them down for each one of those values, but know that each one is going to be in milliequivalents per liter. So sodium is about 12 inside the cell, 140 milliequivalents per liter in the interstitial space, as well as 140 in the blood plasma. Now, generally, you should know that the interstitial space and the blood plasma, which are both considered extracellular fluid, they generally have very similar concentrations of solutes, except for protein, which we'll get to that. Potassium is going to be 140 inside the cell, and then 4 milliequivalents per liter in the interstitial space and in the blood plasma. So lots of sodium outside the cell, and then lots of potassium inside the cell. If you remember in Bio 32 with the action potentials, and we talked about them a little bit with the heart chapter in Bio 33 as well. But having lots of sodium outside the cell allows for depolarization, with sodium rushing into the cell to allow us to reach the maximum voltage for our action potential, while having a lot of potassium inside the cell allows for repolarization, which is the efflux, or the moving out of potassium out of the cell to bring us back down towards our resting membrane potential. So sodium for depolarization, sodium into the cell, and then potassium for repolarization, which is potassium out of the cell. So that kind of gives us reasoning why we have these values here for sodium and potassium. So that's just those values, and we'll talk about other values as we go along in this lecture. But one thing to know is that there are some times when fluid can actually escape these three compartments. So this is where these are the only spaces that we should have fluid in our inside of our cells, in between the cells, and then in the blood plasma. Now, sometimes that fluid, if we have too much fluid, it can actually escape into other areas. And in that case, and I guess I'll put this up here, in that case, we call that abnormal accumulation of fluid, third spacing. So third spacing is the abnormal accumulation of fluid, which many times takes place in a body cavity. And when you have fluid inside of a body cavity, not in one of these three spaces, not in the intracellular space or the interstitial space or in the blood plasma, if it's not in these three compartments and it's somewhere else floating around in a body cavity, this is where that fluid can't carry out normal function. So we don't want that fluid in escaping to a body cavity where it can't actually carry out any normal functions. And so this kind of third spacing, this escaping of fluid into places where it doesn't belong, can be due to burns, can be due to edema, which we've talked about before. We've talked about edema with congestive heart failure. We've talked about lymphedema when it's a lymphatic problem. Also ascites we talked about with cirrhosis of the liver, or with kwashiorkor and lacking protein in your diet, as well as post-surgery, or in case of certain trauma, that fluid can escape into places where it doesn't belong. Okay. So that is third spacing. So I'm gonna erase these notes. So if you need to write them down, press pause and then keep going. Let me erase that milliequivalents per liter. So we're gonna do a little review on solutions, which you should have learned in B32 back in chemistry, but you're gonna match the term with a definition. Solute is either a homogeneous mixture of two or more components or substances, or a dissolving agent or medium, or the substance that is dissolved. So in the class, I would ask you which one is which, but a solute is going to be the substance that is dissolved. So it's a substance that we're going to be dissolving in the solvent to become a solution. The solvent is the dissolving agent or medium, what we're dissolving that solute in, which is usually water. And then when we bring that solute and solvent together, that makes a solution, which is a homogeneous mixture of two or more components or substances. So know that a solute plus a solvent makes a solution. Okay. And that should be reviewed. Next, we're going to talk about electrolytes. Electrolytes, and this is all in your lecture notes, but electrolytes are chemicals that separate electrically in fluids. So that includes salts like sodium chloride, where in solution, the sodium separates from the chloride. So we have a positive sodium ion and a negative chloride ion. Or in cases, well, we're going to talk about this next, but acids. So acids are electrolytes that release a hydrogen ion specifically. So when the electrolyte, when it separates chemically, if one of those ions is a hydrogen ion, it's considered an acid. And so what we're going to talk about here, the difference between a strong acid and a weak acid. So a strong acid is a chemical or an acid that completely dissociates into its ions. You cannot separate it anymore. So that includes hydrochloric acid. So hydrochloric acid is made up of a hydrogen ion and a chloride ion. So when it's in solution, it dissociates completely. Those are the only two parts it has, and it separates from that combination. So it's hydrochloric acid together, HCl. But as soon as it hits solution, it separates into its hydrogen ion and its chloride ion. And because that's as far as it can separate, we consider that a strong acid, okay, where it completely dissociates into its ions. Now, a weak acid is when it only releases, in this case, is one of two of its hydrogen ions. And so I'm going to write this out. So carbonic acid, which we've talked about a lot, and I'm sorry, it doesn't allow me to do the, or at least I don't know how to do the subscripts and the subscripts at all. So you can see it should be H with a little 2 and then CO with a little 3, but H2CO3 actually has 2 hydrogen ions that are associated with it. But when it's in solution, it releases only one of its 2 hydrogen ions. And so because it only releases one of its 2, it doesn't completely dissociate, then we consider it a weak acid. So when carbonic acid separates in solution, you should know this already by the bicarbonate buffer equation that we've been talking about a lot this semester. It separates into an H plus and an HCO3 minus. And you can see that there's still hydrogen associated with that bicarbonate, HCO3 minus, so it doesn't completely dissociate. There's a carbon that doesn't dissociate. There's an oxygen that doesn't dissociate. And so because it does not completely dissociate into its parts, one hydrogen is still left on that bicarbonate ion then we consider it a weak acid because it has two hydrogens to give, but when it's in solution, it only releases one out of those two ions. So it's considered a weak acid. Versus hydrochloric acid, it only has one hydrogen ion, but it does release that when it's in solution. So weak acid versus strong acid. A strong acid completely dissociates versus a weak acid does not completely dissociate and only gives up partial of its hydrogen ions. And in the case of carbonic acid, it's only releasing one out of its two hydrogen ions. So it's considered a weak acid. Okay. Next is non-electrolytes. So non-electrolytes are those chemicals that do not dissociate in solution. They are going to retain their chemical structure. They do not change at all. So you can see there this image here, where here's a spoonful of sugar. Helps the medicine go down. Sorry. But this spoonful of sugar here, C6H12O6, when we put it in solution and dissolve it to make some sugar water, that C6H12O6 stays together. It does not dissociate at all. It retains its chemical structure, and so we call it a non-electrolyte because it does not dissociate. Other examples of chemicals that do not dissociate in solution that we've talked about, lipids are one of them that do not dissociate. Creatinine, which we talked about, that kind of creatinine, which tells us a little bit about kidney function. And then urea, which is a nitrogen waste product of protein breakdown. All of these are considered non-electrolytes because they do not dissociate in solution. Okay, so you should know the difference between an electrolyte and a non-electrolyte. So osmolality is the number of solutes dissolved in 1 liter of water. We use a liter because that's generally how we measure fluids, but it's also appropriate to say the number of solutes dissolved in 1 kilogram of water. It's the same measurement, or at least it equates to the same mass. So either one is fine. So what we want to talk about as far as osmolality goes, this tells us osmolality is going to influence the net flow of water. So it's going to tell water which way to move. Okay. One thing you should know is that the osmolality, we talked about how inside the cell, sodium is 4 milli or 12 milliequivalents, sorry. And then potassium is 140 milliequivalents. Well, if you add all of the solutes in the intracellular fluid, it's going to equal to 300 milliosmoles. Same thing for the interstitial fluid. We said that it's 140 milliequivalents per liter for sodium outside the cell, and 4 milliequivalents per liter of potassium outside the cell. So in that interstitial fluid, if we added up all of the solutes, including sodium, including potassium, we'll talk about calcium and protein and all of that as well here in a second. But all of those solutes will equal to 300 milliosmoles. And then if we go to the blood plasma, all of those solutes that we find in the blood plasma will also equate to 300 milliosmoles. So it doesn't matter what the chemical makeup is, each compartment is going to be slightly different. But we did say that the interstitial fluid and the blood plasma are going to be very similar, but they all have their differences. However, the total amount for osmolality is going to be the same. The total concentration for each one of those 3 compartments is going to be 300 milliosmoles. So now we're going to talk about osmotic power in electrolytes versus non-electrolytes. One thing you should already know, this is review from B32, and we talk about it all the time, is that water moves and I think this is already in your notes as well, but water moves according to osmotic gradients. And so that means that it's going to move from an area that has a lesser osmolality to a greater osmolality. So it goes from areas of low concentration to areas of high concentration. So it moves from lesser osmolality to greater osmolality. And when we say osmolality, we can also use the term concentration. So we're going from areas of low concentration to areas of greater concentration when it comes to water, Because when we say that it has less osmolality, so it's less concentrated, that means there's more water in this area. When we're talking about an area that has a greater osmolality and it's more concentrated, that means there's less water there. So water is going to move in a way that makes the water balanced on either side, or really the concentration on either side. So if it's really low concentrated on one side and highly concentrated on the other side, water is going to move towards the higher concentration side to be able to dilute that concentration to make it equal with the other. Okay. All right, and so one thing about osmotic power, so electrolytes versus non-electrolytes. So non-electrolytes, when they are in solution, when they're in a fluid like water, they only contribute one solute to that solution because they don't dissociate. They stay in their original form, C6H12O6, so glucose stays glucose. So we're only contributing one solute per molecule of glucose that goes into that solution. An electrolyte, though, because it does dissociate in solution, so you're making multiple parts when it goes into a fluid, electrolytes contribute two or more solutes into a solution. So hydrochloric acid, you're not only putting hydrochloric acid in there, you're putting a hydrogen ion into that solution and a chloride ion in that solution. So you're putting two solutes in there. Same thing with carbonic acid. You're putting a hydrogen ion and a bicarbonate ion in that solution because they are going to immediately dissociate or separate. And so anytime you add an electrolyte to a solution, you're really adding two, maybe more solutes to that solution. Versus a non-electrolyte that does not change, does not dissociate, you're only adding one solute per molecule for that particular chemical. And so the question I usually ask here is, which has a greater ability to cause fluid shifts? Is it going to be an electrolyte that separates and adds more solutes to that solution? Or is it going to be a non-electrolyte that stays the same and only contributes one solute to that solution? So I'll give you a second to think about it. And if you're thinking electrolytes, you are correct. Because if we're adding more solutes to that solution because we're adding electrolytes and they're separating and adding more solutes than a non-electrolyte, and that's going to attract more water to that area because it's more solutes. You're adding two or more solutes for every molecule of that electrolyte that you're adding. And so more solutes means higher osmolality, which means water is going to be more attracted to shift in that direction. Okay, so I hope that made sense. And then last thing on this slide is that for electrolytes that do dissociate in solution, we measure them in milliequivalents. The reason why we measure them in milliequivalents is because that takes their electrical charge into account, whether it's a 3+, 2+, whatever it may be, it takes that into account as it influences osmolality. And non-electrolytes, because they don't dissociate in solution and we don't have to take into account their charge. Non-electrolytes, because they don't have charges, they're not chemical like that. They're not made up of ions in that way. But non-electrolytes are going to be measured differently. They are measured in milligrams per deciliter, or the same thing as milligrams per 100 milliliters. Okay. So the next thing that we're going to look at is we're going to look at those three fluid compartments and what they look like as far as concentration goes. So you can see the extracellular fluid, which is in blue and red. That is the extracellular fluid, the interstitial fluid being blue, the blood plasma being red. That is its own compartment together. Those two make the extracellular fluid. And then the intracellular fluid is in yellow. So you'll notice across all of these, for sodium, the extracellular fluid, the blue and red bars are pretty equal, while the intracellular fluid is quite different. Because we said before that inside the cell, it's about 14 milliequivalents per liter. Outside the cell, it's about 140. For potassium, still blue and red are about equal. We said that potassium inside the cell is outside the cell is going to be 4 milliequivalents per liter, and we can see that here. Versus inside the cell, it's going to be at 140. So for sodium, we have lots outside the cell and less inside the cell so that we can have that depolarization and the influx of sodium. But for potassium, we have lots inside the cell, less outside the cell, because having more potassium inside the cell allows for the efflux during repolarization. Same thing for calcium, pretty much the same with the red and blue bars. Magnesium, pretty much the same with the red or blue bars. Bicarbonate also, chloride also. All of these are going to be pretty similar between the blood plasma and the interstitial fluid, except when it comes to protein. This is the only place where it's going to be different. We said before that our intracellular fluid has lots of protein because our ribosomes inside the cell make lots of protein for us. We said that our blood plasma also has lots of protein because our liver makes blood proteins for us, like globulins and fibrinogen and albumin that we talked about during the blood chapter. But in that middle space, in the interstitial fluid, we really shouldn't have too much protein. There should not be protein in that middle area because we don't want fluid shifts into that middle area. So, and there's really no method or reason for having protein in that mid compartment. The only time that we have proteins in that compartment to a high degree is if there's an issue, either an issue with filtration or capillary damage, stuff like that. So should not have protein or very much protein, little to no protein in the interstitial fluid, but we will have a good amount of protein in both the intracellular fluid and in the blood plasma. So make sure you take a look at your lecture notes. Your lecture notes should have a breakdown of the 3 fluid compartments and what the concentration should be for sodium, potassium, calcium, chloride, proteins, and bicarbonate. So just make sure that you know those values. We're going to use those values, especially for bicarbonate in the fluid balance 2 lecture. So just make sure that you are paying attention to that. I'm not sure if this was corrected yet. So make sure you look at the total concentration for the intracellular fluid and interstitial fluid and the blood plasma. They should all say 300 milliosmoles. I know one semester the plasma had a little bit of a typo and was missing the MO for milliosmoles, so just make sure that they're all equal across. All right. And so again, we already talked about this and why we shouldn't have protein in that interstitial space. We know why we have protein in the other 2 spaces. So this is an image we've already looked at. So just make sure that you now use your lecture notes and that table there to reference those different concentrations. All right, so a review question. We're going to take each one of these chemicals one by one. I think I just gave you the answer, but we're going to take each one of these things one by one, and you're going to tell me whether it's a chemical, an electrolyte, or an acid. So first, glucose, is it a chemical? Yes, anything that's made up of elements is going to be a chemical. And so, but is it an electrolyte? No, it's not an electrolyte. Electrolytes are those things that dissociate in fluid. Glucose does not dissociate in fluid or solution, and so it's not an electrolyte. Is it an acid? Well, no, acids also dissociate in solution. So if it's not an electrolyte, it's not going to be an acid. Carbonic acid, is it a chemical? Yes, anything made up of elements is a chemical. And so is it an electrolyte? Yes, because it does dissociate in solution. And is it an acid? It says it right there in the name, carbonic acid. It is an acid. However, it is a weak acid. So make sure you know that because it has 2 hydrogen ions to give, but when it dissociates in solution and only gives up one of those single hydrogen ions. Next is HCl. Is it a chemical? Yes, it is a chemical. Is it an electrolyte? Yes, it is an electrolyte. It does dissociate in solution. Is it an acid? Yes, and it's a strong acid, hydrochloric acid. It only has a hydrogen ion and a chloride ion as a part of its chemical makeup, and when it is in solution, it completely dissociates, gives up the hydrogen, gives up the chloride, and so it's considered a strong acid. Sodium chloride. Is it a chemical? Yes, of course it's a chemical. Is it an electrolyte? Yes, it is an electrolyte. It does dissociate in solution. But is it an acid? No, it's not an acid. It's actually a salt. The only way a chemical or an electrolyte can be an acid is if one of the ions that it gives off when it separates is a hydrogen ion. Sodium chloride does not have a hydrogen ion. It's just a sodium and a chloride ion. This is a salt. So it's not considered an acid. Urea is next. Is it a chemical? Yes, it is. Is it an electrolyte? No, we said it does not dissociate in solution. It stays as urea in solution in a fluid. And so if it's not an electrolyte, it cannot be an acid. But make sure you know the difference between an electrolyte and an acid when it comes to the final exam. I think I'll stop there and then we'll have a smaller little chunk of lecture on fluid movement across compartments or among compartments.